vaccine technology, delivery, and improving antigenicity. Adjuvants stimulate an immune response. Inactivated virus particles or purified proteins often do not induce the same immune response as replication component intuitive preparations unless mixed with a substance that stimulates the early inflammatory response. Such immunostimulants are called adjuvants. Their development has been largely empirical, although as our understanding of the various regulators of the immune response increases, more specific and powerful adjuvants are being discovered and employed. Vaccine researchers can optimize a vaccine by using different combinations of adjuvant and immunogen to induce a protective immune response. Adjuvants act by activating early intrinsic and innate defense signals, which then shape subsequent adaptive responses. These immunostimulators function in at least three distinct ways: by presenting antigens as particles, by subsequenting antigen at the site of inoculation, and by directly stimulating the intrinsic and innate immune responses. The latter effect occurs when adjuvant mimic or induce cellular damage or alter homeostasis, sometimes called danger signals, or when they engage intrinsic cellular defense receptors. Adjuvants vary in compensation, from complex mixtures of killed microbacteria and mineral oil to lipid vesicles or mixtures of aluminum salts. Some adjuvants, like alum, or you could say aluminum, are widely used for human subunit vaccines such as the papillomavirus and hepatitis B vaccines. Others, such as completely foreign adjuvant, are used only in research. This adjuvant is extremely potent but causes extensive tissue damage and toxicity. Two of the active components in Freud's adjuvant have identified an myrmidopeptide and lipid A, both potent activators of the toll-like receptor 9 pathogen recognition protein. Less toxic derivatives, along with sapiens and lineopolymers, are clustered hydrophobic and hydrophilic monomers, are promising as far as alternatives. Now, for example, a new version of hepatitis B virus vaccine, Heplosav B, contains a new adjuvant that appears to promote a stronger immune response than the long-established vaccine, which used aluminum salts. At present, vaccines are delivered by a limited number of methods, typically the traditional hypodermic needle injection or oral administration. Delivery by injection has disadvantages, as discussed previously, and therefore improvement of the administration of vaccines is an important goal of manufacturers. Other methods under consideration include new emulsifying particles and direct injection of fine powders through the skin. Oral delivery of vaccines can effectively stimulate IgA antibodies at mucosal surfaces of the intestine and inducing a more systemic response. Genetically engineered edible plants that synthesize immunogenic viral proteins represented a new attractive approach to designing potent and cost-effective oral vaccines. Transgenic plants expressing viral antigens can be developed, or plant viruses with genomes encoding immunogenic proteins can be used to infect food plants. Species such as tobacco, rice, maize, potato, alfalfa, lettuce, tomato, carrot, peanut, and soybean are used as hosts for gene introduction, typically via nuclear or chloroplast genome recombination. The choice of the plant species and technology determines the vaccine administration route because some plants can be consumed only when processed, and heat or pressure treatments may destroy the antigen. Cereal crops such as attractive for subunit vaccine production because vaccines produced in seeds are stable over long storage periods. However, oral vaccination, by whatever methodology, is not always possible because the enzymes produced in the oral cavity, coupled with acidity of the alimentary tract, destroy many vaccines because they can induce a protective response. Use of plant cells is an exception because the plant cell wall is resistant to acid environments. Vaccination patients who are already infected with the viruses that cause persistent infections or that are reactivated from latency present special problems. One approach to resolve an established infection is via immunotherapy. Immunotherapy is a strategy in which the already infected host is administered antiviral cytokines, antibodies, or lymphocytes over and above those they already possess. Immunotherapy can be administered by introduction of purified compounds or of a gene encoding the immunotherapeutic molecule. An intuitive virus or DNA vaccine can be modified to additionally synthesize cytokines that stimulate a desired immune response or skew a host response toward a particular cytokine profile. If an intuitive vaccine is supplemented, care must be taken to ensure that the subsequent host response does not lead to undesirable outcomes, including increased virulences, persistent and/or pathogenesis of the vaccine strain. An experimental immunotherapeutic drug vaccine to treat Ebola virus post-exposure compromised 3 chimeric monoclonal antibodies. During the 24 Ebola virus outbreak, ZMapp was mobilized quickly for use in humans, having only been previously tested on animals. Despite a later plan to perform a clinical trial, the epidemic warned and the trial closed early, leaving the trial underpowered to give a meaningful result. A relatively new promising immunotherapeutic strategy against human immunodeficiency virus type 1 is the use of broadly neutralizing antibodies, or what they call BNABS. BNABS are antibody cocktails that neutralize multiple human immunodeficiency virus type 1 strains. They do so by binding conserved epitopes in the virus particles embedded in protein regions that are essential for viral reproduction. The first BNAB was identified in 1990. This antibody recognized the 6 amino acid cluster at the top of the GP120, the viral protein that binds the cellular receptor. While this BNAB was not useful clinically, it is important proof of principle and led to the identification of a second BNAB, also specific to the GP120, that affected 17 of 24 human immunodeficiency types strains examined. Now, the discovery of these cross-reactive antibody specials, coupled with single-cell antibody cloning, made it possible to produce large quantities of the antibodies for study. Trials are underway to determine if administration of these antibodies can reproduce the risk of human immunodeficiency virus type 1 infection. Broadly neutralizing antibodies may also be promising immunotherapeutic avenue for other viruses that mutate rapidly, including influenza viruses. Historic successes with vaccines for smallpox, measles, polio, and other viruses, combined with promising new formulations such as the virus-like particle preparation that protect against hepatitis B virus and papillomavirus infections, have transformed modern medicine. However, our lack of progress with other viruses, including the failure to develop an HIV type 1 vaccine, invariably successful annual influenza vaccines, remind us that viral infections and immunity are intricate and poorly understood processes. Differences in a host immune response to individual viruses and the difficulty in developing safe and infectious vaccines without a complete grasp of how immunity works in each case pose significant roadblocks to progress. We have learned that intervening in any complex host-parasite interaction sometimes produces unanticipated effects. Indeed, we frequently find out how little we know when vaccines are tested in the real world. Formulations that worked in a laboratory, animal model fail in a natural host. Sadly, even when vaccines are available, people may refuse to accept them, and or societies may not be able to afford them. The issues, which are most about public perception and education, determine how effective a vaccine campaign will be. As viruses do not respect country boundaries, such as issues best addressed by international organizations. Despite these hurdles, new technologies and ideas are being introduced at breakneck pace. As infectious disease experts work to test whether these new approaches might vanquish long-standing pathogens. For example, Efforts are underway to create a universal flu vaccine that affords protection against all influenza virus strains, regardless of the virus subtype or viral genetic drift. Different strategies, including targeting a highly conserved epitope in the surface proteins and development of antibodies against the conserved stalk domain of the hemoglobin, are currently being discussed as promising therapeutic tools against influenza virus infections. Because of the conversations of the stalk domain, these antibodies are able to broadly neutralize a wide spectrum of influenza virus strands and subtypes. These candidates could be developed into universal influenza virus vaccines that might obviate the need for annual vaccination. It is noteworthy and somewhat humbling that most successful vaccines on the market today were developed empirically. Think back to a high risk experiment that Jenner attempted, and how this was based more on a hunch than on actual scientific evidence. As we learn more about the molecular mechanisms of antiviral immune defenses and the epidemiology of infections, we can hope that the development of future vaccines will be based on logic and established virology principles rather than fortuitous guesses.